do so. At this time, then, I'd like to welcome P.B.Hatzinger from CB&I Federal Services, and he is going to give us his final project summary on his CERDIP project number ER-1607 that was on new approaches to evaluate the biological degradation of RDX in groundwater. Paul, I'll turn it over to you.
Okay, great. Thanks, Andrea.
Sure.
Welcome, everybody. So for this project, our project team was pretty diverse. It included Bella Chu at Texas A&M, J.Hawari, formerly at the NRC Biotechnology Research Institute, who's now in some capacity at McGill University, Neil Sturchio is now at University of Delaware, Steve Fallis, Naval Air Warfare Center, China Lake, and then myself and my colleague Mark Fuller here at CB&I. So this project was in response to a 2007 statement in need. It was a 2008 New Start project, the objective of which was to develop an improved understanding of the biological degradation of nitroamines in the environment. And in terms of our technical objectives, we sort of mirrored that objective initially. We wanted to sort of develop a little better understanding of RDX degradation, and we sort of approached this through a series of questions that at the time we started this, I think were not well answered. You know, is RDX degradation occurring? Under which conditions? Sort of what are the key pathways, and can we measure specific metabolites? Which organisms are responsible? How are these organisms using RDX in the environment? Can we effectively quantify or verify degradation processes in situ? And how can we possibly better promote degradation and or enhance rates? So we had four main technical objectives, and this is what I will ultimately present results upon. The first was to analyze indicator metabolites of RDX at sites. And we started out looking at methods to preserve some of these different metabolites. We collected groundwater samples from, I think it ended up being a total of 44 wells from several different sites. And then we looked for different types of metabolites that, that you can get during RDX degradation. And really the key question here was, is biodegradation occurring in aquifers? And if so, what are the key pathways that are occurring for RDX? Our second task was then to look in the laboratory at RDX degradation under differing geochemical conditions. So we collected aquifer and groundwater samples from two sites, and these were two sites that we sampled during task one. It turned out one was Picatinny Arsenal, and the other one was the Naval Surface Warfare Center in Dahlgren, Virginia. And I'll talk primarily about the Dahlgren data today. But from those samples, we prepared microcosms, mesocosms, and columns, and I'll talk about a subset of those data. We looked at things like alternate carbon sources, and we looked at RDX degradation under a suite of different geochemical conditions or electron accepting conditions, like denitrifying, iron reducing, sulfate reducing. And at the end of these, we looked at, the degradation of RDX, metabolites produced, which geochemistry appeared most beneficial. We looked at microbial communities, and then we did compound-specific stable isotope analysis of carbon and nitrogen in RDX. The key question here is really, under what geochemical conditions does RDX occur? Our third main task was to apply a technique called stable isotope probing for identification of RDX degrading organisms. This is a really interesting technique where you add isotopically labeled compound in an environment, and in this case we added either isotopically labeled nitrogen or carbon within RDX. Bacteria then incorporate this into their DNA, and you can separate out the labeled from the unlabeled DNA, and then subsequently run a whole series of different potential techniques to look at microbial communities and also to identify organisms that have actually incorporated the carbon or nitrogen. So this is a really sort of interesting technique that directly ties the ability of an organism to incorporate some carbon or nitrogen from a given compound to that compound being in the environment. You don't really have to isolate the organism, but you can see which organisms are responsible. And that was really our question, which organisms are actually carrying out RDX degradation in the environment, and how are they utilizing this compound? And our fourth main task was then to evaluate the biological fractionation of isotopes. This is using compound-specific isotope analysis, where you're looking at sort of an increase in stable isotope ratios as bacteria degrade RDX. And this included some method development work. We sort of developed a newer, improved method for analyzing nitrogen and carbon isotopes in RDX. We did quite a bit of work looking at pure cultures to really try and understand in the laboratory how carbon and nitrogen are fractionated, so how much these different bacteria actually enrich carbon and nitrogen isotopes during biodegradation of RDX. And then we also did some work with mesocosms, where we incorporated soils and groundwater from sites and then tried to see if we could see isotopic enrichment. For this presentation, I'm going to focus mainly on some of the pure culture data. We want to see, can we use this to then quantify RDX degradation in the environment purely by using stable isotope analysis. So as we tie all of this together then, our first main task is sort of broken down into kind of validating preservation, collecting samples, and then looking for metabolites just to try and see if biodegradation is occurring. Our second task is a series of microcosms, mesocosms, and columns, looking at different geochemical conditions. Task 3, looking at bacteria. Task 4, kind of the stable isotope work, and then the whole thing sort of ties together into, sort of improving our understanding of RDX degradation and trying to optimize in situ remediation. So as we move into the metabolite analysis, I wanted to include a couple of kind of the main pathways that have been identified for RDX degradation. And this is what we believe is the primary aerobic pathway of degradation. And what's important here is we see it's a double denitration early on. So RDX, we see release of nitrite twice with we see nitrate circled there. Then as we move forward through these proposed intermediates, one of the things that we come up with is a compound called NDAB, 4-nitro-2,4-diazobutanol. And this is a compound that probably in the environment occurs only from degradation of RDX, so it's a nice indicator metabolite. And as we move further along, we see things like formate and ammonia, and which can be important but have multiple sources in the environment. And it's important to note that NDAB, in all the work people have done with aerobic RDX degraders, NDAB's actually only been detected a couple of times in the environment. So it's very interesting to go out and look for this in a bunch of different groundwater aquifers to see if it's occurring. And I should say you don't see it by your typical 8330 explosives analysis. So that's also part of the reason potentially that people haven't observed it is, we're not looking with the right methods. Now, anaerobically, degradation of RDX is quite a bit more complex, and I've kind of simplified this into two pathways. The top pathway A was that which was originally determined by McCormick et al. Back in the 80s. And essentially we see a nitro reduction. So the nitro groups in RDX sequentially become nitroso groups, the MNX, DNX, TNX. Then we get ring cleavage, and we get a variety of different common products, things like methanol and nitrous oxide. But the MNX, DNX, and TNX should be, again, good indicator metabolites, because you're not going to find those occurring in the environment other than from degradation of RDX. In the second pathway, our B pathway, there's an early ring cleavage of RDX. I don't think anyone is still quite clear sort of how this occurs. This is quite simplified, and I think there may be multiple reactions that may occur early on as this ring is cleaved. But one of the products that's formed is methylene dinitramine, called MEDINA or MEDINA. And again, this is a fairly unusual compound and probably one that you're only going to see from degradation of RDX. And as we move further down the pathway, we see things, again, that are more common in the environment, but could also be indicative if there's relationships in terms of, if you see degradation occurring in these increasing, things like nitrous oxide and formaldehyde. So I'll just review a subset of our preservation data. One of the things we were concerned about early on, and this again was in conjunction with J.Hawari, was no one had really looked at preservation of some of these metabolites in the past, and and what methods might be appropriate. Often samples are acidified, for preservation, and sometimes the pH is increased, but there's a lot of different things that can occur. But if we look at graph A in the upper left for MEDINA, we see that at a typical groundwater pH, this is a pretty transient compound. We see in the red, sort of the red line there at pH 5.5, this really doesn't last very long. Does not last a little longer at very low pH, and then at a high pH, very alkaline pH, it does seem to be a little bit more persistent. So we started looking at potential preservatives for the compound, and actually Jalal had done some work with marine degradation of RDX and noticed that in seawater there was some preservation of some of these compounds. They were found to persist a little bit longer. So one of the things we looked at was just sea salt. And in fact, if we look at graph B in the upper right, essentially what we have here in the different symbols there are just increasing percentages of sea salt at two different temperatures. So the closed circles are 23 degrees, and the closed symbols and the open symbols are at 4 degrees. But one of the things we see is that the combination of low temperature and sea salt tends to be very protective of MEDINA in the samples. At warmer temperatures, you still see a lot of degradation. And if we look down below, when we use optimal conditions, so 10% sea salt at 4 degrees in groundwater, there's very little degradation of MEDINA in those samples over a period of nearly a month. And that's whether the samples are filtered or left unfiltered and unsterilized. Now moving a little further with this, the other compound that was of great interest was NDAB. And if we look at graph A, we see that at pH 5.5, say a typical groundwater pH, NDAB is pretty stable anyway, even at 40 degrees, which is good to see. It does seem to degrade at lower and higher pH. So if one were acidifying samples or increasing the pH a lot, you might need to be concerned. But again, as we started thinking about the sea salt as a preservative, if we look at graph B in the lower left, we see that at 4 degrees C in groundwater, the sample, with or without sea salt, was remained, but the sea salt certainly didn't impact that. So if you added sea salt to a sample, you could preserve both MEDINA and NDAB with no problem. Similarly, in the upper right, we looked at a lot of preservatives for formaldehyde in samples, and it turned out that sea salt worked fine, as did methanol or ethanol. And MNX, DNX, and TNX in the lower right are very, they're very stable compounds anyway, and the sea salt didn't impact those, which was also good to see. Another test that we ran, and this will be the last of sort of the preservation methods, but was just looking at actual site water and site water under anaerobic conditions. So if we, these were some samples from Picatinny Arsenal that we had previously added cheese whey to, so the water was quite anaerobic. And we found out that, in fact, NDAB, again, was very stable there in the presence or absence of sea salts, and those are the two graphs on the left. And this included with additional nutrients and cheese whey added. We didn't see a lot of degradation of NDAB over a period of more than a month. Now with MEDINA, if we look at the graphs on the right, the sea salts were protective, not completely, but they were reasonably protective in these environments, and it's either the F is for filtered, the NF is for not filtered, and then you have SS is sea salts, so plus or minus. In the absence of the sea salts, we see quick degradation, and it didn't matter whether the samples were filtered or not. So that indicates it's just an abiotic process. And this does seem to be a pretty labile compound in the environment overall. So then, in terms of our key conclusions, NDAB can be expected to be very stable in groundwater. MEDINA, on the other hand, is very short-lived. And 10% sea salts, along with just cool temperatures, which are typically used 4 degrees, can be used to preserve essentially all of these different metabolites. So you could go out and sample, if you're interested in looking at MEDINA, formaldehyde, along with MNX, DNX, TNX, and RDX, and NDAB. you could add sea salts at 4 degrees C and preserve all of these different samples or compounds and metabolites. So from here, once we had preservation techniques, we went to the field and collected RDX and then sort of looked for some of these metabolites to see what we would find. Just a list of some of the different sites we looked at. They sort of ranged in some different areas of the country. If we look at the map in the upper right, Mass Military, Picatinny Arsenal, Indian Head, and Dahlgren. And then sort of out in the west and southwest, we went to Umatilla in Oregon, Pueblo Chemical Depot in Colorado, and then the Pantex plant in Texas. We sampled for all of these different metabolites that I spoke of, as well as basic geochemistry, metals, and other parameters. And ultimately, we sampled 44 different wells at 7 sites, along with some background wells that had never previously been exposed to RDX, or the groundwater hadn't been contaminated previously, based upon the best knowledge that we had. There were a few instances where some wells were sampled on multiple occasions. And if we kind of look at the lower right, we see that most of the groundwater had a reasonably neutral pH, but we kind of got a good range of geochemistry if we look just at ORP. We had some very oxidizing wells, positive 100 to 4 1/4. Some that were kind of in a lower range, 0 to 100, and then some wells where conditions were anoxic and reducing with ORPs from 0 down to about negative 250. Now, in terms of analyzing for these indicator metabolites, we have this little graphic on the left that just shows, kind of the things that we were looking for by the different pathways. So this is just a summary of the data from all of the wells that had positive ORP and detectable DO. So these are our aerobic wells. Now, we didn't find any NDAB in 6 of the 7 sites, so a vast majority of the wells. We did find some at the Pantex plant, so that was quite interesting in 2 different wells. We didn't see a lot of formate or formaldehyde. We didn't detect nitrite, which we know is released by aerobic RDX degradation, but it's also fairly quickly converted to nitrate or other nitrogen compounds in the environment. No MEDINA was detected. Now, we did see elevated nitrous oxide in some wells, which was quite interesting. And if we look at this graph on the lower right, it wasn't a great correlation, but clearly there was some correlation to RDX. So as we saw, RDX concentration increase, we did see some increased nitrous oxide. Now, presumably this would occur at the very end of the degradation pathway, but it could also be a bit of a red herring in that, nitrous oxide can also be produced from nitrification and denitrification. We didn't follow up a lot further on this, but I think it would be interesting to try and look at what the isotopic characteristics of nitrous oxide are that are released from RDX and see if there's anything very unique compared to what you find during aerobic production of nitrous oxide, say during nitrification, or anaerobic production during denitrification. However, clearly that's a fairly significant effort. But this correlation we thought was interesting to show. Now, we were kind of excited about the detection of NDAB at the Pantex plant. Again, it hasn't been detected very much in the environment. But there was some curious data here in that there's been a lot of different things that were done at the Pantex plant over time in trying to remediate RDX. And curiously, in these wells, this just shows, the four of the wells we looked at, you see they all had a positive ORP. Some of them had nitrate, which indicates, that you've got at least reasonably aerobic or oxidizing conditions. But you'll also note that 2 of the wells also had some of the anaerobic products, DNX, TNX, and they also had NDAB, so that was sort of curious. Now, one of the things we did is we wanted to see if we could isolate organisms, or at least confirm that RDX was degrading aerobically in these wells. So we set up, we collected additional samples. We set up microcosms with and without nutrients, under aerobic conditions, with and without a little bit of succinate. And in fact, over nearly 5 months of running these microcosms of all different types, we never saw RDX degradation under aerobic conditions. And we also did probing for XPLA, which presumably would be the responsible gene that produces the P450 enzyme that carries out this denitration reaction, that was also not detected. So, we didn't have a lot of good follow-up here that suggests that there was aerobic degradation going on. And some work after this done by M.Fuller and Jalal also showed some production of NDAB under largely anoxic conditions, which is kind of interesting. So maybe that's actually what was occurring here. So then finishing up with our anaerobic metabolites, we really didn't see any MEDINA in any of our reducing wells, even with proper preservation. So it's, if it's formed, it probably doesn't last very long in the groundwater to begin with. We did see a little bit of MNX at some wells. We saw some DNX and TNX at Pantex, as I mentioned. It's hard to say if this was just naturally degrading anaerobically because, again, at both of these sites, including Picatinny, there was some previous work done to try and stimulate degradation before we had taken these samples. We didn't see elevated nitrous oxide. We did see some formaldehyde and methanol, and I just show you the graph below where we see formaldehyde as a function of ORP at these sites, and you get some pretty high levels of formaldehyde in some of the groundwater. But we think largely this is not related to RDX degradation. The concentrations are just too high to begin with. It's rather just a function of production from anaerobic fermentation or other processes in the environment. So to wrap this task up then, NDAB is relatively stable in groundwater, but it was not detected other than in a couple of different wells. And where it was detected, there was no XPLA, and some of the MNX, DNX, and TNX was also detected. So, one of the outcomes of this project in the field sampling was that it doesn't appear that aerobic RDX degradation is a particularly prominent mechanism in the field, at least based upon the samples that we gathered and the metabolites that we were able to look at. We did not detect MEDINA in any anaerobic wells. Again, it's a very labile compound. You can see things like formaldehyde and methanol, but they're probably spurious. They're probably produced from other sources. And nitrous oxide is an interesting one and something that may warrant further investigation. And most of these data were published in 2011, along with the preservation methods in Journal of Environmental Monitoring. So moving on to our next task, and I'm going to show sort of a subset of results that I thought would be kind of most interesting to the group. There's other data that are included in the final report for this project. So what we did here is, again, we collected our groundwater and aquifer solids. We did some initial screening just to look at which substrates most promoted RDX degradation. And then we set up some large-scale, what we'll call mesocosms, where we tried to stimulate different electron-accepting conditions. So with nitrate as a predominant electron acceptor, or iron, or manganese, or sulfate, so that we could stimulate different geochemistry and try to understand a little better how RDX degrades in these different sets of conditions. And then again, we looked at metabolites, we looked at microbial community analysis, and some stable isotope fractionation. So the site that I'm going to show data from, again, we have data from both Picatinny Arsenal and Naval Surface Warfare Center Dahlgren. I'm going to focus here on the Dahlgren data. This is a site where it's been around for nearly 100 years as a Navy test range. There's a couple of different areas where there's RDX and HMX in groundwater, and it's fairly typical. It's tens to hundreds of ppb of these at the highest. There's also some perchlorate. That's not particularly relevant to what we do here. But largely the site is aerobic. There's some marshy areas that are anoxic, but it's largely aerobic. And the pH is reasonably low at 5, and sometimes it gets a little bit lower than that even. Now, if we just look at stimulating RDX biodegradation in microcosms, on the left, we tried all sorts of different carbon sources, and for the most part, they were all effective. So we were able to stimulate organisms that degrade RDX with a suite of things ranging from ethanol to cheese whey to glucose. It's not that this is unusual, but we've had other sites, and Picatinny was one of those where this wasn't the case. So you have to be a little bit cautious about interpreting this because it's possible at some sites that some of these electron donors are going to be more effective than others. And for one reason or another at Picatinny Arsenal in New Jersey, cheese whey really was the best carbon source to promote RDX degradation, and many of these others didn't work particularly well. But as I mentioned before, we ran aerobic microcosms over a period of time. We never saw any evidence of RDX degradation in aerobic microcosms, and that's whether we added a little bit of carbon source to try and promote something to use RDX as, say, a nitrogen source or a sole nitrogen source or not. So this, again, sort of further goes with this hypothesis that aerobic degradation may not be all that prevalent in the environment. So from here, what we did is we set up these larger scale mesocosms. You know, each of these got about 500 grams of aquifer solids and 8 liters of water. They were pressurized under nitrogen to keep them anoxic. We didn't set up an aerobic mesocosm because, again, in our microcosms, we didn't see any evidence of degradation. So the first graph I show here on the left, and basically what we were trying to do here is just sort of set up conditions where RDX was degrading, because ultimately we're going to take portions of these mesocosms and then put them into small bottles, and that's where we're actually looking at microbial communities and degradation and metabolites and those types of things. So these bottles were more kind of a longer-term incubation. But one of the things we see here, now RDX is, in each of these is the red X's, that's your RDX. Succinate is the black diamonds. Perchlorate's also on here. But what we see is that under nitrate-reducing conditions, so when we add nitrate to these microcosms, we see no evidence of RDX degradation. And at a point around 200 days, we spiked in a little extra RDX, which is why you see that occur. And we were continually spiking nitrate in here. That's why you see things sort of going up and down from 10 ppm. You know, whenever the nitrate became limiting, we respiked it to really try and establish those conditions. So, you know, one of the take-home messages here is if you have a lot of nitrate, it's possible that that's going to inhibit RDX degradation. There's some different results in the literature, but this is certainly supported by a few papers in the literature that suggest that if you have a lot of nitrate, you're really not going to see RDX degradation. And we didn't see a lot of perchlorate degradation either, which is also consistent, and that nitrate can inhibit that at times. So the next graphs we're looking at on the left, this is iron-reducing conditions. On the right is manganese-reducing conditions. And again, we continually spiked in iron and manganese into these, along with additional RDX. We see on the left, as we're moving along here, we see our amounts of soluble iron increasing, showing that we've established conditions where we're forming ferrous iron. We're getting up to 30 milligrams per liter plus. RDX is degrading. We're respiking it. It's degrading so quickly you don't see those spikes typically. Same thing under manganese-reducing conditions. And this is over a period of only you know, a few months. So we established very good degrading conditions under both sets of iron and manganese-reducing conditions here. And then our other kegs were sulfate-reducing. So we tried to establish good sulfate-reducing conditions. Once again, this took a lot longer, almost a year, continually spiking with sulfate when it ran out, and succinate as a carbon source and additional RDX. But we do get good degradation. We get obvious sulfate-reducing conditions. And same thing in our last keg with, we tried to establish methanogenic conditions just by consuming all of the other electron acceptors. And here we do start to see some production of methane, you know, so at least suggesting that we have established some methanogenic conditions in those kegs. So from here, essentially, those kegs were split up into 1 liter bottles, and the samples were spiked with different types of isotopically labeled RDX. I'll talk about that in a minute for the SIP analysis, but there was another thing that was we looked at some of the degradation intermediates that was both interesting and probably potentially important here. And if we look at the left, so these are our bottles that we respiked with RDX under manganese-reducing or iron-reducing conditions. They got additional succinate, so essentially all the kind of stuff that we were doing in the larger kegs. And one of the things you see in purple there is actually TNX. So under both sets of conditions, we see production of TNX and persistence of TNX over pretty long periods of time. So again, these bottles were incubated. That day 0 was when we re-amended things into the new bottles. And again, we're going another 3/4 of a year for the manganese-reducing conditions. And we have a lot of TNX persisting. If we look on the right, on the top right graph, we see under sulfate-reducing methanogenic conditions, we have very rapid RDX degradation under these conditions, and we don't see much production of these intermediates. And what the intermediates that are produced are transient, and they're pretty much gone in 15 or 20 days. So what this kind of suggests, if these results would carry through to the field, is that you'd like to get to a point where you have dominant sulfate-reducing or methanogenic conditions if you don't want things like these NXs essentially to persist, and typically you don't. You'd like to, you want to make sure those are completely degraded if you're remediating RDX. Okay, so to carry on with this mesocosm study then, our next sort of task was to look at the bacteria that are responsible for degradation under these geochemical conditions. And I sort of showed this graphic before, but essentially we split, as I said, we split these samples into bottles. You incubate with different types of isotopically labeled RDX, carbon or nitrogen labeled, and I'll show you a little bit more about that in a minute. And this really allows you to kind of directly link organisms with the observed activity that's going on in the environment without any prior knowledge of the strain of interest. And essentially what you do here then is you separate out the DNA, and I should say this part of it was, we did all the incubations here in our laboratory and collected all the DNA, and then we send filters down to Bella Chu at Texas A&M, and her students then would take the collected DNA, separate the bands, which is what you see in the middle in that sort of purple gel. You can separate a 14N labeled DNA from a 15N labeled. And then there's a technique where that material is then cut up. So what you do is you put a fluorescent marker on the DNA, and then you use a restriction enzyme, and from the marker down, the DNA gets cut into different sizes, and essentially, that each piece represents a different organism, presumably. There can be some overlap at times, but so presumably each of these different bands, which has to do with a restriction fragment, so if you look at the graph on the upper right, that's just number of base pairs, 82 base pairs through 300 some odd base pairs. These different fragments represent an individual organism. And in cases where you see a band in the 13C DNA fraction but nothing in the 12C DNA fraction, that's a unique organism that incorporated 13C into its DNA. So the molecules that we used for this were specially synthesized at China Lake by Steve Fallis, who's a chemist there, an excellent explosives chemist who can do this sort of thing. And he basically made molecules for us that were either carbon labeled, like we see on the left, so they had C13 in the ring, or we had molecules that just had nitrogen labeled in the ring. We had those that just had nitrogen in the nitro group, and we had those that were fully labeled. So this was, to perform this type of work, you really need these types of molecules. They're not available commercially, so that was a nice collaboration with the Navy to get these made for the project. Now, as I mentioned before, everything from the mesocosms was then put in individual bottles that were then spiked with all of these different types of N15 or C13 labeled RDX. Obviously, this was done individually in separate bottles, so this ended up being a pretty large study under all these different electron accepting conditions. And then we measured degradation of RDX, and at select points when we had most degradation had occurred, we took samples and then ran the process that I just showed a few minutes ago. Now, some of the results, and essentially we're looking at, again, those T-Riflic diagrams on the right. We have number of gene copies on the left, so this can also be quantitative, so presumably telling you kind of how many of these different organisms might be in your samples. And in this case, anything that was just labeled with the sort of the unlabeled carbon or the unlabeled nitrogen is excluded. So we're just looking at bands that were labeled with either 13C DNA or 15N DNA. And in the case on the right, You see that, basically any of the red bars, that was RDX originally labeled with the ring-labeled RDX. Green is the nitro-labeled RDX. The clear bar is the 13C, and the black is the fully labeled. Now, as we look through this, we see a couple of different generalities. One is that there's some pretty high densities of organisms involved in some cases, where it's something like 10 to the 6th per mil or 10 to the 9th per liter. Pretty high. These are pretty high concentrations of some of these organisms. As an interesting example, if we look at the C13, so that's the clear bars. Under every different electron accepting condition, from manganese reducing in the top graph down to methanogenic in the bottom, 4 of these bands occurred. So there were 4 organisms, presumably, that degraded RDX in some function under each set of electron accepting conditions. But there were also a couple of unique ribotypes, for instance, in iron reducing, where you can see a couple of bands sort of here and here that not quite there, but close enough, that really don't occur elsewhere. So again, unique organisms that were occurring under these different sets of conditions. Now, in terms of the nitrogen, there was also something that was fairly interesting overall. If we look at the red versus the green, so that's the ring-labeled versus the nitro-labeled RDX, we see that they don't occur together very often. There's a few circumstances where you see the two, and again, that seems to be predominantly under iron-reducing conditions, sort of in a couple of spots here. But otherwise, they're largely separate. So it suggests that organisms that are using the ring nitrogen and those using the nitro-nitrogen are actually separate types of organisms. Again, fairly interesting. So, good data, and it's also interesting to note how many different organisms are involved. It's important to state that we don't know that all of these organisms are using the parent RDX. They could be using metabolites, and some of them probably are. But again, it kind of shows you that there's a large community of organisms that are involved in this process. Okay, so now cloning was also done and organisms were identified. And this is kind of a separate process from the T-RIFLIP, so they're sort of independent analyses that are done. But what's interesting here, there's a lot of organisms involved, but quite a few of them in the lower right here fall into Clostridia and into a genus of organisms called Bisulfosporosinus. Those had never been previously associated with RDX degradation, but clearly there's a variety of different isolates under a variety of different electron accepting conditions that are associated here with RDX degradation. The one other place that these have been observed was in our work at Picatinny Arsenal. So 2 different sites in very different areas we found this organism. So perhaps there's, this is an interesting and unique organism that should be identified further. Unfortunately, to my knowledge, no one has isolated an organism of this genus that actually degrades RDX in any way. People have isolated the sulfovibrio, which is a related organism, but not this genus. And also, if we look at the top sort of yellow area, we see there was a variety of other bands under manganese and sulfate-reducing conditions. And they group closely with some of the Pseudomonas species. And M.Fuller and J.Hawari have previously studied some of these organisms that have what's called the Zen A and B gene that can degrade RDX. The other thing that's important is you see there's kind of a mix of different electron accepting conditions that fall under these 2 groups. But if you look in the literature, this Bisulfosporosinus as well as the Pseudomonas species, they have very diverse metabolism. So, in the case of the latter, on the lower right, these organisms can grow with a variety of different electron acceptors. So it does make some sense that they in fact can occur under all these different sets of conditions. Now, in terms of the 15N, we kind of see the same thing. Which is pretty interesting as you look at these different cultures. We see these Clostridia, Bisulfosporosinus, and a lot of different organisms under these different electron accepting conditions that group there. And in this case, we're looking at nitrogen. So apparently this group of organisms used both carbon and nitrogen in RDX or some metabolite of RDX during growth, which I think is quite interesting. And again, we see sort of this predominance of Pseudomonas species also. So there's sort of, despite the fact that there's a lot of organisms, they're kind of grouping together in a couple of different classes here. So then in terms of some of the conclusions, RDX degradation was inhibited in the presence of nitrate. And these pathways and metabolites may differ depending on the dominant electron accepting conditions. I think that's very important to think about as you move forward with trying to stimulate RDX degradation. There's a lot of organisms involved, but they are sort of grouping. They may be sort of different species within a couple of different genuses in some instances. And some organisms that weren't shown to be involved in RDX degradation, what we historically rely upon is you have to isolate an organism and then you study that organism. Here, we didn't isolate anything, but we found these groups of organisms that were involved that no one had previously isolated. So this is certainly more complex than one thinks when one looks at the current literature on RDX degradation, where there's a growing number of organisms that have been isolated, but certainly not hundreds. We're still in the probably 10 to 20 range. So I'm going to finish up then with task 4, and this is again using stable isotopes, but in a little bit of a different method. In this case, we're not trying to label bacteria, but we're just trying to see naturally what bacteria do in the environment as they degrade RDX. And just as a little background, as bacteria degrade many compounds, they degrade a compound that has a heavy element. So in this case, if we look at the graphic, red in the top are molecules of RDX that have N15 incorporated into their structure. The degradation of those occurs at a somewhat slower rate than what occurs for RDX with N14 incorporated. So ultimately, as time goes by, what you end up seeing is you start to see disappearance of RDX and an accumulation of N15 RDX in the parent material, and you would get an accumulation of your lighter label. So, say N14, in this case, we're looking at MNX as a product. And that's called isotopic fractionation, and it can be a very powerful technique in the environment. It's more and more widely used to document contaminant degradation. It hasn't been widely used for RDX, but that's that's one of the things we started to work on here. As part of task 4, there was some method development work. This was done at the time. It was at University of Illinois, Neil Sturchio's lab. He's since moved to the University of Delaware. But essentially, by this process, it's a isotope ratio mass spec that's coupled to a gas chromatograph. So what happens is you concentrate your sample. So you can get, say, a liter of groundwater. You concentrate that through an SPE column into a very small volume. You elute it in acetonitrile. And then you inject that onto a GC IRMS. And essentially what happens is we run through a GC phase where everything gets separated, and then materials are combusted into gases. And what you do is you measure the isotopic composition of either the nitrogen that's produced or the carbon that's produced as nitrous oxide or CO2 respectively. And that's how you ultimately get your stable isotope ratios of carbon and nitrogen in RDX. And the other nice thing here is that you only need about 10 micrograms of RDX per analysis. So you can analyze sites where you have pretty low RDX concentrations. If you at 10 ppb or lower, you can get you can get isotope values for carbon and nitrogen, again, just by concentrating these materials. And we've since, through an ESTCP project, have developed a field method also to concentrate these samples a little bit further. So what we did here is we looked at a number of pure cultures, and we looked at cultures that degrade RDX either through the aerobic pathway, which we see at the top here, this double denitration I showed before, or a couple of the different pathways, the ring cleavage or the pathway through MNX, DNX, TNX, as we see as pathway 2 and pathway 3, and we looked at how they fractionate RDX isotopes. And what we found, if we start with the aerobic pathway, we found consistent nitrogen isotope fractionation of about -2.5 to -3 per mil. And what we're essentially looking at here in each of these different graphs is on the x-axis, it's just basically the fraction of RDX that remains. So from the right to the left, that gets less and less. It's just a natural log function. And on the y-axis, we're looking essentially again at a natural log function, and it's the isotope ratio of your material at any given point in time over that in the parent. So in this case, your N15 versus N14 ratio in RDX versus that was originally in the parent as a function of time. And when you plot these out, you can get these nice, when the relationships hold, you can get these nice linear graphs and fits, and that's that epsilon value, or the slope of that line, is your basically a fractionation factor. So that's how you can compare among different strains. And this is a reasonable fractionation. You can see that the standard deviations are low. You're fairly consistent among two different strains that degrade RDX by the same pathway, so that's all very positive in terms of using this method in the field. We didn't see very much carbon fractionation, but that's consistent with what we know about this pathway, because we're really not impacting a carbon. We're impacting a nitrogen molecule in the denitration reaction. And I should say that little red line in the upper left is actually in the wrong location. It should be at a nitrogen-nitrogen bond. It must have gotten moved around as I was working on the slides. But you wouldn't expect a lot of carbon fractionation, so this is also consistent with what we know about the pathway. Now, for the ring cleavage pathway, where we're breaking a nitrogen-carbon bond, we see here that we got really large fractionation of nitrogen, about -13 per mil. We also got some carbon fractionation, which also would be expected. So again, if degradation is occurring by this type of pathway in the environment, you expect to see large isotopic shifts and shifts in both nitrogen and carbon ratios. Now, the third result was for these pathways where you presumably see MNX, DNX, and TNX, so the sequential nitro reduction pathway for RDX. Here you're breaking a nitrogen-oxygen bond initially. We saw, again, very significant nitrogen isotope fractionation, which again is great for a field study. But we also saw some carbon isotope fractionation, which really wasn't expected because, again, there's no carbon bond involved, and typically you can have secondary isotope effects, but you wouldn't specifically expect a significant carbon isotope effect here. But as we compared our results here with all of the metabolite data, what we ended up finding, was that for a lot of strains that we thought went just through this MNX, DNX, TNX pathway, in fact, they were not producing a lot of those metabolites in some instances. In other instances, for strains that we thought were primarily ring cleavage strains, they were producing some of these metabolites. So I think one of the things that this tells us, in fact, is that during anaerobic degradation, some of these strains may be using multiple pathways to degrade RDX. And this is not necessarily novel. You can have multiple enzymes induced that can handle RDX in different ways, and in particular, we know it can be used as a nitrogen source, a carbon source, an electron acceptor. There's a lot of different potential uses for RDX. But this is kind of telling in that we may have a good number of organisms that are doing this in different ways in the environment as well, and the isotope data supports that. If we kind of lump all this together then, if we look at our aerobic degradation, we can expect to see nitrogen isotope fractionation of about -2.5 per mil with a range of about -2 to -2.8 under aerobic conditions. Under anaerobic conditions in the bottom with all these different strains, our average was about almost -11 per mil in terms of nitrogen fractionation, -4 per mil per carbon. There was a little more of a range there, which again suggests that you may have different ways that these molecules are attacked, different enzymes involved, but again, very good stable isotope fractionation. So one would expect that in the field, if degradation is occurring, you very well may be able to see it using compound-specific isotope analysis. So I'll sort of wrap up, and again, I didn't cover everything we did in this project, but I tried to hit some of the highlights. The report's available for anyone who would like to read a little further. But essentially, we developed methods and tried to better evaluate the potential for RDX degradation in the environment. We looked at metabolites. We did stable isotope probing to identify bacteria. We did some compound-specific isotope analysis. And the application of these methods suggests some different things. Pathways of RDX degradation may differ under electron-accepting conditions. You may want to promote certain type of electron-accepting condition. Aerobic degradation really may not be all that prevalent in the environment, despite the fact that we've had a lot of lab studies of aerobic RDX degraders. A lot of different organisms may be involved, particularly under anaerobic conditions, including some that were not previously associated with this activity, and that the CSIA may have broad application for both aerobic and anaerobic degradation of RDX in the environment. Our transition plan, we're currently validating this method through ESTCP, the CSIA method. Neil Sturcio can offer this on a per sample basis at University of Delaware. We've already done a lot of presentations and continue. We have several publications. I'm still participating as a trainer on the ITRC Environmental Molecular Diagnostics team. That team is a few years old now, 4 or 5, 10, I'm not sure. And we do cover CSIA and SIP as part of that training. Some of our future research needs: develop a better understanding of the fractionation of carbon and nitrogen at a mechanistic level in RDX, maybe evaluate stable isotope data for some of the other things released from RDX, further assess pathways in the field, apply the CSIA method at field sites, and to continue to sort of look at the role of T organisms, see if there could be indicator organisms that start showing up at a lot of different locations, such as, interestingly, the Pseudomonas and Desulfospora sinus that we have identified in this study. We have 5 publications that are either already published or in preparation and pretty much ready to be submitted. And with that, I'll conclude the presentation and take any questions.
Thanks, Paul. Are there any questions?
Yeah,
well, I guess I'll go ahead, Marvin.
Paul, this is Marvin. Stellar presentation, Paul. Just a real quick question. What does Neil charge for a stable CSIA analysis? What is the going rate for something like that? I know years ago it may have been a good deal higher. What are we looking at now?
Marvin, it's an excellent question, and it's certainly specific to the compound. Now, for RDX, there's yet to be any commercial analysis requested. We're probably within a day or two of submitting sort of a large paper that covers all of the pure culture data that I showed you, and kind of for the first time we'll really get this method out. So perhaps that'll change. And I don't know what he'll charge. For the perchlorate isotope analysis that he's done for many years, it's relatively expensive, but there's an awful lot of sample preparation work that accounts for most of the cost.
Right.
In this case, that's minimal. It's really just taking a water sample through solid phase extraction, putting it into acetonitrile. If we look at commercial labs doing things like TCE, you're in the hundreds of dollars per sample at this point. I'm trying to remember. It wasn't that long ago. I had some TCE analysis run, and I want to say that we did TCE, cis-DCE, vinyl chloride, and I think we were in the several $100s for the whole suite of those, and I think it was something like $250 or $300 if you just wanted TCE. So, I assume we're going to be in that range somewhere, Marvin, if there is some demand for the analysis.
Well, we certainly want to see this deployed on a commercial scale, and anything we can do to get the cost reasonable would be great. Thank you very much, sir.
You're welcome, Marvin. Thank you.
Paul, this is Hans. I was curious about the geochemical factors, and what is going on under aerobic conditions, and what do you think under iron and manganese reducing? Do we have to drive this to deeply anaerobic basically to get good treatment, or what?
Again, really good question, Hans, and I don't think we didn't really expect that, to be honest with you. And the thing that doesn't quite make sense if you kind of follow through this is that a lot of the organisms were fairly similar under those different sets of conditions. Yet, in terms of the metabolite data, pretty clearly we stalled out at TNX under iron and manganese reducing, and quickly those metabolites were gone under sulfate reducing conditions. Now, I suspect that, I think as the years have gone by, I've kind of come to believe that a lot of these organisms will continue to coexist no matter how you drive the conditions in the environment. We're continually now seeing aerobic co-metabolic organisms being found in anaerobic environments and vice versa. So I think you have a large mix there. When we've done things like added emulsified oil at the same Dahlgren site, our ORP didn't go incredibly low, but we also didn't see a lot of buildup of metabolites. So, clearly we were getting down into a realm where degradation was passing through those metabolites. We did see certainly some iron produced, but there was also a lot of evidence that we were down in sulfate reduction. So, I think you're getting to a point where all of these different processes are occurring. I think two of the things that are fairly clear, though, from what we're seeing is that aerobic degradation doesn't occur that commonly, at least in groundwater aquifers. We haven't found much evidence for it. And also, I think nitrate can be a significant issue, and part of this may be the fact that some of these organisms want to use RDX as a nitrogen source, and in the presence of nitrate, they're going to use the nitrate first. So if it's a nitrogen limited situation that causes them to turn on an enzyme to use RDX, and you've got plenty of nitrate around, as we do in many different areas of this country in aerobic groundwater, it's possible that those two factors are contributing to the fact that we don't see a lot of degradation going on at these higher ORP conditions.
Great, thanks. Really interesting.
Any other questions? Okay, if not, thanks very much, Paul, for taking the time to provide the debrief today. Really appreciate it. I know it's some extra work, but it's very helpful to the program.
Well, thanks, Andrea. Thank you for the invitation, and thank you for everyone who took an hour to attend as well. And if there's any further questions, please give me a ring.